Hi everyone, what's going on? Welcome to a new episode of the Frontier of Knowledge podcast. Now today I was joined by Dr. Dave Carter, who is a lecturer and researcher at Oxford Brookes University. His main focus is related to cancer and some of the cell mechanisms related to it. Today we also discussed some of the challenges that we're facing currently in developing new treatments for cancer, and why some of the current treatments we have now are basically ineffective. We also talked about some of the new or potential new mechanisms related to how people are screened with cancer and how that's going to change over time. So sit back, relax, and I hope you guys enjoy the podcast. If you do like today's podcast, please hit that like and subscribe button and enjoy. Hi everyone, welcome to the Frontier of Knowledge podcast. Today I'm joined by Dave Carter, who is a researcher and lecturer in Oxford Brookes University. So Dave, if you want to introduce yourself and tell us a bit about what you do.
Hello. I'm Dave. I'm a reader in biomedical science at Oxford Brookes University in the Biological and Medical Sciences department, and so I have an active lab that is interested in trying to understand various things, including how cells talk to one another, particularly during a stress response, using a type of molecule called an extracellular vesicle, and I'm interested in how those things occur during normal conditions, so during normal stress responses within an organism, but also how that can be applied to understand diseases a little bit better, like cancer and other diseases. So, yeah, that's what I do.
Nice. How long have you been doing this for?
So I've been doing this particular job here for about 9 years now.
Oh, nice.
No, 8 years. Since 2009 was when I arrived. So yeah, 8 years, just over 8 years. Yeah.
That's good. What did you prior to that?
So before that, I was at a university called Cranfield, where I also had a lab there for about 2 years. And before that, I was a postdoctoral researcher at Oxford in the Dunn School of Pathology. And then before that, I did my PhD at Cambridge University between 2000 and 2003. And before that, I did my undergrad at York University.
Okay.
In 96 to 2000, with a year out in industry, which was done at the Sanger Centre, where they did the Human Genome Project.
Oh wow.
Which, yeah, it's quite cool.
That's awesome.
Someone on the T-shirt, which is one of my happiest achievements. Yeah. So awesome. I'm from up north originally.
Nice. So, what kind of stress responses do you see in these kinds of molecules when they're undergoing specific kinds of disease like cancer or...
So there's different kinds of stress responses that occur. There are stress responses that occur during normal condition when things get a little bit out of kilter and you have to then change in order to kind of get back to the normal. So like that, a normal homeostatic response, as you know. But cancers in particular have a number of different stresses that they undergo. One is that they, just by their very nature, they become stressed because they grow faster. In theory, they grow faster than they're supposed to be growing, and so the environment around the tumour becomes a little bit corrupted, and the cells are growing too fast, which means they can't get enough oxygen, for example, which means that the whole area becomes a bit hypoxic, which means it's lacking in oxygen. And so they become a bit stressed because of that. You also get a slightly more acidic environment around those cells because of various reasons, and so the cells aren't particularly happy. But also they then become stressed when you try and kill them.
Okay.
So if you try and kill them with things like radiotherapy, with chemotherapy, then they can become quite stressed, and actually also normal cells can become quite stressed. So if you bathe the body in a horrible chemotherapeutic drug, then in theory lots of cells around the body become stressed because they're exposed to this drug. And so we're interested in how cells talk to one another during those kinds of stresses, because we think that these things called extracellular vesicles, which are essentially like a kind of little tiny bag that cells release, and they carry contents, they carry cargo. So cells,
logistics.
yeah, they're just like little bags. So cells put, I mean, they're much more smaller than a cell. So they're like a small proportion of the width. They're very, very small, and so cells put things into these bags. And it used to be thought that these bags were a way of cells getting rid of rubbish.
Okay.
Like little bin bags, really.
Mm.
And then a few years ago, 15, 20 years ago, people realised that actually they're not just bin bags.
Mm-hmm.
Cells are not just getting rid of this stuff. Other cells can then pick up those little bags and actually delve inside and open them up and see what's inside and use what's inside. And so when cells do that, they can respond, and they look at the message that's inside of these vesicles, and they can respond. And so what you put into these bags is in some ways, as a cell, is in some ways a message to the world which tells them about how you're feeling right now. I mean, it's obviously it's a
bit more.
I'm using a metaphor, but essentially, your current state is in part reflected in the content of these vesicles.
Okay.
And so, if you become stressed, you put something different into those vesicles, and so when other cells take up those vesicles and look at the contents of those vesicles, they respond in a now different way. And so, in a sense, what we're finding is that, in a sense, they're alerted to some kind of stress or danger, and they respond by altering their own state to try and compensate for that.
Oh, I see. So as a result, does that have a different effect on metabolism? So inflammation for one?
Yeah, it can have effects on metabolism. So vesicles can affect metabolism. What we're finding is that essentially the cells, the recipient cells, these are the cells that take up these vesicles. When cells take up vesicles from other stressed cells, they respond essentially by trying to become tougher. So
trying to adapt.
So they're trying to adapt. Okay. So there's an adaptive response, exactly. So the cells adapt and they're harder to kill. And so what we're finding is that if you then stop that communication from going on, then it means that overall the tumour's a little bit easier to kill.
Okay. So is there a way of actually delivering a drug where the stress response isn't released, and then therefore you can get rid of the tumour a quicker response?
Yeah, that's what we're trying to do.
Okay.
So the data that we have at the moment, I mean, so we've just got a paper that's close to acceptance at the moment where we essentially showed that is that if you block that crosstalk, not by blocking the release, although you could also do that, but actually by blocking the uptake. Okay. So by blocking the uptake of these bags, of these vesicles, you can, in theory, stop the recipient cells, or as we also call them, bystander cells, because they're not the ones necessarily targeted by the stress, but they're the bystanders that pick up the signal. Okay. They become a little bit easier to kill. You prevent this adaptive response, so they're a bit easier to kill. And if you do that if you view the cancer cells as a population of cells, then if you are stopping this crosstalk, then on the whole they're a bit easier to kill. Okay. So we're wondering whether that might have implications in drug treatment during chemotherapy and radiotherapy, that it could lead to better, more effective treatments for cancer. It's
a really interesting mechanism, that is.
So it's quite new. I mean, no one's really looked at some people have also looked at similar things, but essentially, we've thought of a different way in which this applies, and we're trying to see whether adding these drugs that block uptake could be a way of.
So how do you actually stop the cells uptaking the information?
There's various ways. So there's various routes by which these vesicles can get inside of cells, and actually it's not 100% clear what the most important pathways are. They seem to be able to take many of these different routes. Okay. And so it's not obvious how to necessarily block them completely, but there are various drugs that you can use that block these endocytic pathways, as they're called. Endocytic because endo bringing things inside, and cytic, which is related to cells, so it's cells bringing stuff in. So there's lots of endocytic pathways by which vesicles can get in, and we can block them with certain drugs, which are some of them already FDA approved. So we're trying to go for ones that basically are already in the clinic to try and, it's more convenient and it's potentially more effective.
Okay, it's really interesting. Is there any dietary kind of ways of blocking these kinds of uptakes at all? Was it specifically drug related?
There might be. Okay. I don't know off the top of my head of any.
Coming from a nutrition background, I think it'd be quite interesting maybe if there was some sort of drug-free or maybe nutrition way of blocking these the uptake. I think that'd be quite interesting. I think
maybe.
I guess we have to do research and find out.
Yeah, it's a question of trying to test different compounds. I mean, off the top of my head, I don't know of any specific nutritional compounds that would do that. Just trying to think. No, yeah.
Okay.
I don't know.
Well, fair enough. I guess we'll have to.
But yeah, but it could be easily enough. It's easy enough to test it. You just have to
find the right, I guess, kinds of foods and run trials.
Try screening of some kind.
Yeah. Okay. Now I want to ask you a few questions on different types of drugs and how they work and different other kinds of compounds.
I might not know the answer, but yeah, okay.
I think I bet both of us. I think you'll probably want more information than me. So I've always been interested in looking at the effects of like cannabinoids, the cannabinoid system, on the effect of cancer. And some of the papers I've read, they're finding some interesting results, but other papers have not found any results at all. And I just wanted to check, what were your thoughts on using cannabinoids as a treatment
for cancer?
For cancer, yeah.
I don't know anything about it.
Okay, fair enough.
I don't know anything. I've not looked into it. I don't know anything around that. I mean, my knowledge of cancer treatment is based on more standard, I guess, treatment. So I know a bit about some of the other therapies that are coming through, like the kind of immunotherapy type options, which seems to be quite promising.
How do they work?
So immunotherapy type approaches, most of them work by trying to get the immune system to recognise the cancer as foreign. Cancers, when you get a cancer that forms and progresses and you become ill with a cancer, one of the things that the cancer has to do or has to have done in order to have arrived, if you like, for it to be bothering you, is that it has to learn how to avoid the immune system. So normally a lot of cancers, when they form in the early stages, they actually get snuffed out, if you like, by the immune system, and you never find out about it. Now there's plenty of evidence that actually the tumours that do progress are the ones which have learnt how to avoid the immune system. So if we can basically get the immune system to learn how to recognise these specific tumours again, then we can get the immune system working for us and getting rid of the tumour for us, rather than have to just send in nasty drugs that kill everything. We just re-educate the immune system that this mass that's growing inside of the body is actually a foreign object, and so it then gets rid of it. So that's one of the big areas of research at the moment.
Is that more of a signalling issue? So say if when the cancer first forms, the immune system will recognise it as a foreign. Over time, does the cancer then adapt to the signalling mechanisms of the immune system?
They can be signalling. I'm slightly out of my area, so I couldn't tell you the exact details of how these things happen. But yeah, I imagine it can be potentially signalling. Could also be to do with the antigens on the surface of the cells. Could be subversion by the cancer of the immune cells by sending them signals. For example, I was talking about extracellular vesicles earlier. There's evidence that vesicles are involved in immune function, and so there's some evidence that cancer cells can send vesicles to these cells involved in the immune system that essentially can give them the bum steer, if you like. They kind of act as some sort of decoy or system for shutting off the immune system, at least stops them from recognising the tumour. So it's slightly out of my area, but basically there are a variety of mechanisms by which they can do it. So we're trying to re-educate their immune system.
Okay, nice. Now, are some cancers more prevalent in terms of the vesicles when they excrete these the information you said? Are some cancers more prevalent? For example, like, are they more able to form lung cancers or liver cancers, or does it completely depend on the formation of it?
So there's kind of some different questions in there, I think. So different tumours can put out different kinds of vesicles, and different tumours can put out different amounts of vesicles, yes. And those vesicles can affect the function of the cells around them and help the tumour to grow, an interesting example of that is a recent paper in Nature, which was actually one of my favourite papers. It's really cool. So what they've shown is that, for example, if you have different tumours, some different tumours have different propensity to metastasize to different locations.
Okay.
Many tumours metastasize, and in the end, what tends to kill you is the metastasis, not the original tumour.
Okay.
So it's when it spreads and starts to get to that late stage that bad things happen. So actually trying to understand metastasis when it goes to a new site in the body is one of the key things to try and understand. And so they showed in this paper that different kinds of tumour cell that have a different propensity to go to different locations, or their tropism, as it's called. So some cells have a tropism for going more specifically to the lung, some prefer to go to the liver, some prefer to go to the brain. And what they showed in this paper is that actually if you precondition the body, and this was done in animal studies, so think of those what you may, but so if you prepare the body with the vesicles of that cell, of that tumour, then it seems that helps to prepare the site for the arrival of the tumour. It's also known as the pre-metastatic niche.
Okay.
So essentially what the vesicles do is they seem to, at least in this context, what they showed in this paper, is that they act as kind of emissaries, if you like. They go ahead and then they lay down the ground, if you like, for receiving some tumour cell potentially. And so different vesicles from different cancers, from different tumours, have slightly different contents, and essentially those contents can also act as an address. So by having a slightly different addressing system on it, it goes preferentially to a different place. And actually what they showed is if you take, for example, the vesicles produced by a lung tropic cell line, so a tumour that prefers to go to the lung, and you put it into the body, and then you just prepare the body with these lung specific vesicles. You then take a tumour that normally would love to go to, say, the brain, and you put it into the animal, and actually this cell line no longer seems to prefer to go to the brain anymore. It now prefers to go to the lung, and that's because we think that part of what's deciding where the tumour would prefer to go is down to actually what are the vesicles that they're releasing. And the thing about that is that can potentially give us a lot of information. It can help clinicians and doctors and so on to find out in advance where to look for these tumours and where to try and stop the tumours going to, because the thing about these vesicles is that they're released into all body fluids, and so because the tumours are constantly secreting these vesicles into whatever space they're sitting in, into the bloodstream, into the urine, or whatever, we can potentially tap into that. So remember that these vesicles are floating around. If we were to reach in and grab a load of them, we could then try and look at them, and it's more complicated than that, of course. It's quite technically challenging because these things are very small. But if we can actually look at them and then look at their contents, then potentially we can detect the tumour earlier, because we can look for these biomarkers, as they're called.
So better screening.
So we can screen for these. They're biological markers for the presence of the tumour. So we can look in the bloodstream for these vesicles that scream there's a tumour around, because this kind of vesicle isn't normally present in normal people. It's only present in people who have tumours. And so we can detect the tumour earlier, potentially, but also if we get more into it and we look at what is the content of this vesicle, potentially we can then say, okay, well, this vesicle, the vesicles from this particular tumour seem to have contents X, Y, and Z, which means that they're more likely to, let's say, cause resistance, drug resistance, or maybe they're more likely to cause metastasis to the brain, or maybe they're more likely to cause, whatever it is. And so actually we can start to decipher a little bit about the behaviour and tell a little bit about the behaviour of the tumour based on what's in those vesicles. Now, we're a little bit way off that, because it's technically very challenging, and we don't fully understand what the messages mean yet. We have to do the groundwork experiments to get a whole bunch of different tumours and get the vesicles from a whole bunch of different tumours and analyse them and look at them and then correlate what is it about the content of these vesicles, and how does that compare to what happened to the tumour? And then we can start to make connections, and we can start to say, oh, well, look, every time you see this protein present, the tumour tends to metastasize to the brain, or every time you find this protein present, then you see that the tumour is drug resistant. So then once we understand what that code means, we can then start to make predictions about prognosis, and also about treatment.
Okay.
So, for example, the ideal would be able to find proteins present in them that tell us, okay, well, this patient's more likely to be resistant to this type of therapy. So in that case, let's skip all that unnecessary treatment, and let's go straight to the second treatment. So that's kind of the hope for this type of technology.
Okay.
But it's a way off, But
Okay, that's really amazing how you can use something as small as a vesicle to find out exactly how tumours are going to behave and stuff. That's really interesting. In theory. So what's the next step? So once you've finished writing your paper, would the next step be to actually do it in a lab and then do it clinical? Or?
Yeah. So with all of these kinds of experimental basic lab findings, you have to then get preclinical models. So there's any kind of, like, for example, for a treatment, for a new kind of treatment, you then have to go through long trials to try and figure out, actually, is this something that works? Is it something that works in animal models? Is it something that works in early human trials? Is it something that works better than current drugs? And the problem is that actually lots of the drugs that you use, because the tumours are very, very different. Let's say, for example, if I had a liver tumour and you had a liver tumour, chances are our liver tumours are very, very different, actually. They're classed as the same thing, but actually they're very, very different.
Well, in terms of genetic level?
In terms of the genetic level, the mutations that cause my tumour are different to the mutations that cause your tumour. And so probably, anyway, in fact, no, definitely. So the mutations may be the same, but even if they were the same, then our genetic background is different.
Oh, I see.
Your genome is your genome, my genome is my genome.
Okay.
So the way that my tumour responds to any given situation will be different to the way your tumour responds to the same situation. So we have to then figure out, do these drugs, for example, work in everybody, or more likely, do they work in some people, and if so, which people and why? So it's super, super complicated, and it takes a long time. Most drugs take like 10 to 15 years to get to from initial stages to actually being in the clinic. So if I made some huge, amazing discovery today, then it'd be another 10 to 15 years before
it's actually significant enough to in the lab.
I mean, sometimes under certain circumstances that can be accelerated, but it's quite rare. Usually you have to go through the due process, and it takes a long time. In terms of biomarkers and using these vesicles as like a window into the patient to see what's going on with the tumour. Same thing. I mean, if you're going to start, it takes a long time to figure out what that code is, and what you see, what that means for the patient. But again, those kind of trials take a long time, and then to actually use it as a diagnostic test, you need approval for that kind of thing. So for diagnostic tests, again, you have to go through long trials to check, how good is the test, does it work all the time, some of the time, so on.
Wow, that's really interesting stuff there. Would it be possible, in the example you made up just now about us having the same tumour but having different genetic backgrounds, would it be possible to maybe get a swab of the tumour that is growing, and then maybe perhaps look at the genetic level where the mutation occurs? And so do you think there'll be some sort of correlation if we had, I don't know, maybe 100 people with the same tumours, seeing what is the most frequent type of tumour, and see whether, in terms of genetically, whether they're very similar or not?
Yeah, people are doing that.
Oh, really?
Yeah. Okay. So there are quite a few studies looking at the genetics of tumours, and you can sequence a tumour, like you can sequence the DNA in a tumour, but also bear in mind that even within a tumour, there are lots of different cells with different genomes.
Okay.
So a tumour evolves, it changes over time. So you get a series of mutations that occur one after the other.
Oh, I see. It's a cascade. It's not just one mutation. That's it.
No, it's several mutations usually, and then so you get a series of mutations over a period of time, and it takes years. By the way, it's like a long, long time. So usually, when you get a tumour, when you go to the doctor and present with a tumour, then that tumour started probably 10 years ago.
Wow.
20 years ago sometimes.
So I think so. Then so the best thing would be to get regular checkups, and I guess just to check the screening, just to so you don't start growing a tumour in.
Well, I mean, geez, I might have one in me now. Who knows? But the thing is that you just don't know. And catching a tumour early is still the best way to beat the cancer. I mean, honestly, trying to treat it is very when it's very advanced. It's very hard to treat it. There've been excellent advances in treating cancers on the whole. Like the overall, say, 10-year survival rate has doubled in like the last 30, 40 years.
Wow.
But that's for some tumours, not for all tumours. Some tumours are still horrifically bad. Like so pancreatic cancer, lung cancer, these are really, really, really bad, and they, the survival rate for those is still very, very low, and it hasn't really changed much over the years. And the reason for that is that they're very once they form and they appear, they're very, very hard to treat. And for many of those tumours, and actually all tumours really, the earlier you catch them, the more likely it is that you can beat them. And that's why biomarkers and trying to catch them earlier using the signatures that are in the blood, for example, is one of the big areas of research. But yeah, what was the question again?
So yeah, just the different tumours.
My train of thought.
No, that's all right. No, it's good. I'll keep going into this quite deeply. So I was reading some papers, and they were looking at how tumours also receive a large blood supply and nutrients. Would it be possible to reduce the tumour size by completely cutting off the nutrient blood intake? Yeah.
So there are drugs called angiogenesis drugs. So angiogenesis is the formation of blood vessels to feed the tumour, and yes, there are drugs which cut off or attempt to block the blood supply to the tumour. The other thing is that newly formed blood vessels so even if the tumour has a blood supply, it's probably quite a recently formed blood supply, and actually recently formed blood supply, if you like, is slightly different to a stable blood supply that's been around for a while, and so you can target that also with specific drugs. And so yes, you can, but the problem with it is that one of the things about tumours is that when you cut off their blood supply and they become hypoxic, so they start to run out of oxygen, they also become more aggressive.
Interesting. So they know they're kind of being attacked in like a weird way.
In a sense, so they respond to it in that way. I don't know if it's that they
consciously know that I'm being attacked, time to destroy this person.
but there are mechanisms in place that mean that, generally speaking, hypoxic tumours are more aggressive tumours. And so and a hypoxic tumour is more likely to metastasize to somewhere else. So if you cut off its blood supply, then
it's come back with a vengeance.
It could come back with a vengeance. And so you probably need to try to attack different modalities of the drug, not just cut off its oxygen, but then also go for other things, cut off its lungs, but cut off its legs at the same time, kind of thing. So just doing the one is not always effective, and it can be more effective in some tumours than others. It depends.
Cool. I think I've got time for one more question, and then call it a day. So, what is the most effective drug treatment there is for cancers, or does it depend on the person itself, or does it actually depend on the actual type of cancer it is?
It depends on both. So over years of trial and error, we know that some drugs work better than others for certain types of tumour. And within that, certain drugs work on certain individuals and doesn't work on other individuals. And so if you treat someone with a drug, some of them will respond and some of them won't. And actually that's one of the big areas of research is trying to understand, by looking at an individual but also sequencing the genome of the cancer of the genome, or the cancer genome of the individual, I should say. So by looking inside of the tumour at the genome, looking at the tumour, looking at the genome of the individual, we'd like to be able to do that, read the genomes, and then just say, right, okay, we've read your genome, this is the best drug for you, this drug should work, this drug won't work. That's called pharmacogenomics, essentially, or pharmacogenetics, pharmacogenomics, depending on how you do it. But so that is essentially trying to do personalised medicine, to look at people's genomes and give them the right drug that will work for them. Again, there are examples of that kind of thing coming out now, but it's an area that's rapidly expanding.
Okay.
So it's not one drug for all types.
Yeah.
And there's different kinds of drugs. So, for example, there are some drugs that cause cross-links to occur. So by cross-links, what I mean to say is you get these molecules that bind to the DNA and form kind of crosses between the different bits of the DNA. And there are some which add on big, bulky blocks onto the DNA, for want of a better word. There are some which block the mechanisms by which the DNA untangles itself during cell division.
Yep.
There are ones which block the things called microtubules. So microtubules are basically like part of the skeleton of the cell, if you like, the structural skeleton, and they're needed to separate chromosomes from each other during cell division. And if you interfere with them, it means that the chromosomes can't separate, and if the chromosomes can't separate, the cell can't divide, and if the cell can't divide, the cell dies. And because cancer cells need to divide to keep going, we can potentially target that. So there's and for reasons that we don't really know, some types of drugs work better than some cancers, and we don't know why necessarily. Wow. I mean, there is research into that, so we have some ideas about why some drugs are better than others. But and I just to sort of end by saying actually that it's actually pretty archaic. Current treatments are still, I would argue, pretty archaic, considering that we think we're in the 21st century and it's all pretty amazing. Actually, a lot of chemotherapy itself mostly is just pretty bad, because what you're doing is you're killing cells that are dividing.
Which are also healthy as well.
Which can be also healthy, which is why often your hair falls out, because you're killing hair follicles, where you get problems with blood cells because you're killing bone marrow. So there's lots of reasons why chemotherapy is actually it's
kind of outdated.
It's very very outdated. I mean, I hope one day we're going to look back and think,
what was that going on there?
Chemotherapy is like
dinosaurs.
It's practically like bloodletting. I mean, it's just so archaic. But there are a whole bunch of therapies on the horizon. Because the thing is the only way in which we can target cancer at the moment is because the cells are rapidly dividing. They're dividing faster than other cells, and only slightly fast. I mean, not it's just that's the basis of the separation from most of these treatments, is that these cells are dividing. And if we target all the cells in the body that are dividing, then we'll have some side effects, but at least the tumour's gone. But the tumour always evolves, the tumour changes, the tumour mutates, and when it mutates, it more often than not comes back. And the reason it comes back is because you can get lots of different mutations. Some of those mutations will have no effect, and so that cell that mutates will carry on. It will just either die or it will just sit there not dividing. But by chance, sooner or later, a cell will mutate, which now has the capacity to block this drug. This particular chemotherapeutic, and so it will then start to grow, and at that point there's not a huge amount you can do. Now there's a whole raft of drugs that or potential therapies that are starting to come online, which are actually based on the last 30 years of research. So there's a lot of people who ask, well, what's happening with cancer? Why haven't we cured cancer yet?
Actually, you try and do it.
Well, it's super complicated. It's really, I think people need to understand that it's very, very, very complicated. Understanding how cancer cells work and what goes wrong in cancer cells is a fascinating field, but there's a huge number of different ways in which a cancer can form. So coming back to that earlier point, you can get cancers forming by different mutations. So my liver cancer could be different to your liver cancer. It's different mutations that occur. There are loads of different mutations that can occur, and they all give the tumour different properties. So if I wanted to treat your cancer in the best possible way, what I really need to do is to actually look at your tumour and to understand what are those mutations and how has that caused that cancer to form. And then I need to give you a drug which is specifically tailored to block the mutations or the problems that have arisen in your cells because of those mutations. So rather than just trying to kill everything indiscriminately that's dividing, I want to get in there surgically, if you like, not surgically, but actually precisely using a specifically tailored drug which has been prescribed for you because of that specific mutation. And then what will happen is that mutation, that those cells will be, stopped or slowed down, and then what will happen is, again, a mutation will occur because you can't stop that. And so a mutation will occur and your tumour will come back. But then what we'll hopefully be able to do is look at it again and go, okay, yeah, now you've got this mutation, which means that because that drug is targeting that particular target, that mutation means that now it can't reach its target, if you like.
Yeah.
But there is a way around that. We'll just add this drug instead. And so in time, in I don't know how long it'll be, whether it'll be in our lifetime or not, but it'll, I hope, become a kind of a managed condition, so that but the only way we're going to get to that point is if we can put more money into cancer research, more money into trying to understand how cancer works, and in time we'll get there.
Okay.
But it takes time.
Of course,
people have to be patient, and that's and it's exciting, and it's really, I encourage people to get into cancer research because it's really exciting. But yeah, it'll take time, but we will get there.
Definitely, I completely agree with everything you said there. I think more money needs to be funded in just general all sciences, I think, and that's how you make more advancements with more things.
Yeah, it takes time.
One more last question before I go, if that's okay with you. Would it be possible to look at the genome of the cancer and then alter it which way you can fix it back to its normal state?
So there's another technology which we haven't really mentioned in this, and that's a technology called CRISPR.
Oh, I've heard of CRISPR, yeah.
CRISPR. So CRISPR is an amazing technology which allows you to do what's called genome editing. Okay. So in theory, we can alter a specific DNA sequence within the human genome, and it works beautifully. But the problem is, there are caveats. There are potential problems that it's not 100% fixed yet. It has to go through clinical trials and all the rest of it. In a flask, if you're growing your cells in a flask, yeah, absolutely, you can take the tools to do CRISPR, and you can alter a genome, and you've got a new genome. In practice, it may not be as precise as we want it to be. We need to put in place a technology that is reliable and safe, so we don't know that this thing will be cutting and changing somewhere where it shouldn't be changing.
Otherwise, you might get more cancers forming.
Potentially. But it may well be that CRISPR does come to be one of the ways in which tumours are treated. I think there's even a trial at the moment for lung cancer in China at the moment about using CRISPR to try and cut out the mutations, if you like. So yeah, it is possible. The difficulty in using a technology like that is actually, as much as anything apart from making sure it's cutting in the right place, is getting it to the tumour. So delivering it to the tumour is actually one of the biggest challenges. So delivery of these kind of therapeutics is really, really tricky. Now, if we can overcome that, then in theory, we could have a way of specifically targeting tumour cells. And the thing is that it should be even cleaner, even more specific than most of the drugs, because the way that this genome editing CRISPR works is that it's based on sequences. The technology is based on DNA strands and RNA strands, and so you can put in these RNA strands which recognize specific DNA targets, and because the DNA of the tumor is different to the DNA of the healthy cells, therefore you, in theory, have a surgical knife which you can use to chop out only the genomes that have these specific mutations. And then if the genome changes again and mutates, then in theory we just change the sequence of the CRISPR, of what's called the guide RNA. And so this new guide RNA goes to the new mutation, and so it becomes an iterative process of when the tumor comes back, you just find a new there will be technologies based on that will come out in the next few years.
Didn't CRISPR come out recently as well, a couple of years ago?
Yeah, it's a fairly recent. It's a bit longer than that, but it's a fairly recent thing. It's based on some bacterial. And this is one of the things. This is one of the beauties of studying science, I think, generally, is that when you sometimes study things that you might think are not necessarily applicable to other stuff, but actually, so this is a cool system that certain bacteria have for defending themselves against other organisms. And so on the face of it, there you go. There's a system for bacteria to defend themselves, and actually, when you think about it, you can now apply this technology. And the way it was worked was by modifying incoming pieces of DNA, and so you adapt that technology to a new setting, and yeah, that's why we need to study all of nature in its wonderful varied forms, because
I agree with you. Yeah.
all of these kind of interesting applications that we never thought of. So yeah, it's a really amazing technology, actually.
Oh, cool. Well, I really enjoyed this.
Right.
I think we should do this again, because I have a lot more questions I want to ask you now. I feel like I got more questions out of it.
Okay.
But
well, there's always more questions. That's the problem, is that every answer brings up
Pandora's box.
Yeah, it's an interesting field.
Definitely. Well, thank you very much, Dave.
All right, no
I appreciate your time.
Okay.
Thank you.
All right, thanks.